When I was a fellow, that's when I became interested in gut motility. I had this young patient at Columbia who was 14 years old. Phones were not quite as good back then. And she had chronic intestinal pseudo-obstruction. This is when the gut just doesn't work, and it's got a variety of causes. But I became fascinated by this problem. She was from Ecuador. Her father was a doctor there, and that's why they were able to get some of their care in New York. She was apparently the only child back then, or the only person on home TPN. She would go to the hospital and mix it with the pharmacist. And you can imagine with that kind of situation, she eventually died. I think it was a line infection that killed her when she was just 28. And that really set me off on this path. The enteric nervous system is definitely, without question, I don't care what anybody here studies, the most interesting thing to study. It's a network of neurons and glia in the intestine. There's probably 500 million neurons. It's got all of the neurotransmitters that the central nervous system has. And if you take a piece of intestine out of the gut and put a pellet in it, it will move in the proper direction. And this is guinea pig intestine. But this was observed by Bayliss and Sterling 125 years ago, and they called it the law of the intestine, and that's that local stimulation of the gut produces excitation above and relaxation below, and that's what propels things down the intestine. That's what we refer to as peristalsis. And it regulates everything, not just motor function, but also sensation, secretion, absorption, microbiome composition of the gut, immune state of the intestine, truly everything. And so you can imagine that when there's things wrong in the neurointestinal system, there's a lot of the diseases that can result. All of these are examples of those, whether it's esophageal achalasia or forms of constipation. That patient with intestinal pseudo-obstruction, about a third of cases are due to neuronal problems. Hirschsprung disease is the one that we're going to focus on. That's the easiest one to diagnose. It is simply the absence of neurons in the lower GI tract. It happens in about 1 in 5,000 babies. And usually it's, as many of you know, 80% of the time short segments. So the aganglionic segment, if these clusters are ganglion cells, which are ganglia, which are clusters of ganglion cells, neurons, and glia, the bottom is missing that enteric nervous system. We'll get back to these yellow lines, which is this extrinsic innervation that comes into the gut from the pelvic plexus and other parts, and it gets hypertrophied in the aganglionic part. But that absence of ganglion cells or an ENS in the lower intestine leads to abdominal distension, failure to pass meconium in many children, and a biopsy makes the diagnosis. Some images are showing, it's so interesting. So the biopsy will show the absence of ganglion cells, the nerve hypertrophy, and then we use a deficiency in calretinin fibers to make the diagnosis. It was first described in 1886 by Harold Hirschsprung, but he was very focused on this dilated part of the intestine, not on the narrow part, which in fact is where the pathology lies. That obsession with the megacolon, it was called congenital megacolon for a long time, lasted for decades. And Ladd and Gross, in their textbook of pediatric surgery, back in 1941, said that it is axiomatic to say that no one has ever been permanently cured of this condition. Things changed with Orvar Swenson in this classic New England Journal paper in 1949. And he said, look, it is our contention that the lesion is in the distal non-dilated segment. And then all of the operations started. Swenson divides an operation just to remove the aganglionic bowel and bring the ganglionated bowel down to the anus. Duhamel said that was very hard to do because you were going to injure things around the rectum. So he said, just leave the rectum where it is and bring the ganglionated bowel behind it. And then Suave modified it and said, just leave the rectum where it is, but just denude the mucosa, remove the mucosa of the rectum and pull through the ganglionated bowel. The operation, many people, well, my preferred operation is no longer called the Suave operation. We call it Yancey Suave in recognition of Asa Yancey, an African-American surgeon who developed the operation prior to Suave, but never got the recognition that he deserved. And so that hopefully we're all trying to correct that. Where you incise the mucosa above the dentate line, remove a strip of the mucosa, then go full thickness and start resecting transanally the bowel. You'll see the narrow part here at about 15 centimeters, it dilates, and a biopsy will show that there are ganglion cells there. And these children do very well after surgery, or at least they should. If Hirschsprung disease is the absence of ganglion cells, then removing that aganglionic segment should just cure the patient. It turns out Ladd and Gross are right. Nobody, even to this day, is really permanently cured of this condition, and that's the problem we're trying to address. About a half will ever achieve normal bowel function, and many others struggle with obstructive symptoms or severe constipation, soiling, an increase in stooling frequency, enterocolitis, which is the thing that kills up to, depending on where you live in the world, up to 10% of babies with Hirschsprung disease. In the US, much less, maybe less than 1% die of enterocolitis. But it's just an inflammatory disorder. It happens before the pull-through or after, even though we've removed the aganglionic part. In these studies of multiple prior studies and meta-analysis that looked at adolescents who had Hirschsprung surgery as a baby, only half ever achieved optimal outcome, which is the same bowel function as someone without Hirschsprung. And another series of studies that looked at these children now that they're adults, because the operation's only been around for about 75 years, only half ever achieved normal bowel function. So why is that? If aganglionosis is the problem, why isn't removing that the cure? There's really two fundamental problems. It's that Hirschsprung's is not just aganglionosis. The normal bowel that we think is normoganglionic doesn't work right. There's in one model that we use, there's actually a neurotransmitter. All the GAD2 expressing neurons or the GABAergic neurons are actually missing in the small intestine and the remaining colon. And it's not like you have normal ganglion cells and then all of a sudden they're absent. There's this transition zone that nobody really understands. We have no criteria for how much to remove. We guess 5 centimeters above the aganglionic segment, but we don't understand the transition zone. And in fact, in another series of studies, we found that there's an imbalance of the excitatory and the inhibitory neurons. That balance is critical for normal motility. But if there's too much excitatory or too much inhibitory neurons, then that normal motility can't happen. So the problems continue. The non-relaxing anal sphincter. If you have in a normal person, you distend the rectum with stool and the internal sphincter relaxes. That's the recto-anal inhibitory reflex. That's absent in Hirschsprung disease before or after the operation. So we have to remember that Hirschsprung is not just aganglionosis. And then we have the other problem that what we do is pretty morbid. Removing there's the usual complications of surgery, but removing the rectum in an infant or a neonate, which many people do the operation very early in life now, even in newborns, you can't see the pelvic innervation in that area, that pelvic plexus of neurons that surrounds the rectum. The sphincters are small and delicate, and removing the rectum is a problem. Now, all of a sudden, your high amplitude propagating contractions don't stop at the rectum. There is no rectum. They go right to the anus. So fecal soiling and frank accidents are issues. Anytime you remove some part of the colon, we know that there's this hypermotility problem. You increase the frequency and the amplitude of those high amplitude propagating contractions. And then there's functional constipation. Manipulate the anus in any child, and the pain that that causes, the reminder every day of what was done to them, will just lead them to make the decision that they will never have a bowel movement again. And some of them are very good at that. So can we do better? And the idea here is, can we actually take neurons that are in the colon, the ganglionated colon, grow them in addition, transplant them to the aganglionic region? So instead of doing pull-through surgery, we harvest stem cells and repopulate that segment. It would avoid the need to remove the rectum, and it would avoid all the injury that the operation can cause. So it turns out that we're lucky that there are neuronal stem cells in the intestine. This is a Wnt1 mouse. That means Wnt1 is a protein expressed on all neural crest cells. In the enteric nervous system, both the neurons and the glia are derived from the neural crest. So in this animal, the enteric nervous system is tomato positive. And so it's all red. Why we have neuronal stem cells, and we have them in the adult intestine as well, isn't entirely clear. We know now that they are glial cells. It turns out that a population of glial cells in the intestine have the capacity to become neurons. So they are neural progenitors. And when we look at their chromatin structure, they have open chromatin at neuronal transcription factors. So they are ready to turn into a neuron. They don't have to change their epigenetic profile at all. They just need the right signals inflammation is such a signal. If you cause colitis in a mouse, all of a sudden glial cells will decide that it's time to become neurons. So maybe it's a mechanism for injury recovery, but for us, it's just nice to know that there are neuronal stem cells that we can harvest from the gut. And if we remove the muscle layer, I didn't mention earlier that, remember that the enteric nervous system lives in two layers, the myenteric plexus, which is between this longitudinal and circular muscle, and the submucosal plexus that lives between the mucosa and the circular muscle. The myenteric plexus has a much richer density of enteric neurons. So if we just remove the muscle layer, we can then digest it, culture the cells, and grow them in conditions that are used for central nervous system stem cells called neurospheres. Neuronal stem cells have this ability to grow as floating spheres of cells in the right conditions. And so these neurospheres are what we call our therapeutic product. This is what we need to grow and transplant. And when we grow them in culture, those spheres contain all the markers we want to see for progenitors in the intestine, glial cells. Green here, some mature neurons, they start to differentiate in this condition. And SOX10 and FOX2B, also markers of neuronal progenitors. You can differentiate them in culture, and here in green, they form neurons, and also here in green, glial cells with just the right morphologic features. The question is, do they do anything? We have a mouse where all the neural crest cells express GCAMP. GCAMP is a protein that fluoresces when it's exposed to calcium. So when neurons see calcium, they turn the GFP protein on. And just in the normal mouse, you'll see that there's this intermittent activation of neurons that happens. If we then take those cells, grow them as neurospheres, plate them, allow them to differentiate, and then look at their neuronal signaling, you'll see that not only are they activating, or they're fluxing calcium, there's calcium signaling that's activating the GFP, but it turns out they're doing it in a way where they're actually connecting with each other. So they're signaling to each other in the culture. All right, so this just tells us we can isolate the cells. We have ways to expand them in culture, to grow them, and they actually function as neurons as far as we can tell. So next we have to move to the cell therapy question. This mouse has Hirschsprung disease, so he's going to be our recipient mouse. The neural crest gives rise to the enteric nervous system. It also gives rise to melanocytes, so that's why they have this white coat color. And you can see the megacolon phenotype here. Here's the narrow distal colon, which is the aganglionic part. You see it better here. This is small intestine, a huge cecum, colon, and then right there would be the transition point, and the rest is aganglionic. Our donor cell in this case comes from a mouse where all of the glial cells, PLP1 are green, and all of the neurons, BAF53B expressing, are red. So that the gut in these mice has green glia and red neurons, and we're going to transplant them into this recipient animal. If we take those the intestine from this animal and make neurospheres, you get these pretty little spheres that have both neurons, glia present. And then we transplant it, in this case, right into the mid-colon just by injecting the neurospheres into the muscular wall. After 2 weeks, you can see that the cells, again, green is glia, red is neurons, and they spread beautifully in the intestine. They extend neurofibers around. They express another neuronal marker, hue. And then when we zoom in, we can see that they're forming what we call neoganglia. They're actually forming clusters of cells, which is what the normal enteric nervous system does. And those clusters of cells, again, are glial cells. They have neurons. And they also undergo neuronal subtype differentiation. In this case, nitric oxide synthase, which is one of those inhibitory neurons. Remember that excitation above and relaxation below. The excitation above is cholinergic signaling. Calretinin is a good example of that, and those are calretinin neurons. And the relaxation below are nitrogen neurons, and they express that too. So that's promising, but so we have neurons, but do they do anything? One way to test that is called electrical field stimulation. In this case, we can take a piece of the gut that we transplanted, put it on ex vivo into this device that has 2 pins. And if you just put this cylinder of intestine. The two pins will send or they're electrodes. So they'll depolarize the neuronal membrane when you apply a voltage difference across those electrodes. And when we do that, so if you take a piece of normal intestine and do that, there's no spontaneous activity, but when you apply electrical field stimulation, you get this contractile response. And if you do it in what's called NANC conditions, so you remove all adrenergic activity and cholinergic activity. So you had an alpha blocker like phentolamine, you had a beta blocker, you had atropine, you're getting rid of all excitatory ability. That'll allow you to expose the inhibitory activity of nitrogen signaling, which is that. Hirschsprung mice have this interesting phenomenon that they have this spontaneous myogenic activity. If the gut, that's the baseline of the gut, it has this spontaneous activity. It's only when you add an enteric nervous system during development that you shut that down, and now the nervous system controls the muscle function of the gut. So in Hirschsprung disease, all you see is that myogenic activity, the electrical field stimulation does nothing. But when we transplant cells, we diminish that myogenic activity and we recover a contractile response. We also recover the inhibitory response is very weak in the intestine, always experimentally. So it's there and it's restored, but marginally so in this example. And then you can quantify all these things. And what's important here is only to say that wild type contractile response to electrical field stimulation and the one that you get after transplanting Hirschsprung's bowel are both eliminated by addition of tetrodotoxin, a sodium channel blocker that proves that these are neuronally mediated. Problem is you don't know if it's the neurons you transplanted. It could be that there were some neurons hanging out in the gut. So the way to address that is to move to optogenetics. Now, optogenetics is a method where we can add a channel rhodopsin. This is a channel that you can put onto any cell. In this case, we put it onto, I think we put it onto the neurons, the BAF53 cells. The channel rhodopsin, in response to blue light, will allow an influx of cations, which will lead to an action potential. So we're only going to be able to stimulate cells that we transplanted. So we generated these BAF53 mice that express channel rhodopsin. They're labeled red. So all of these are these neurospheres from that mouse. We transplant them. You can see beautiful extension of neurofibers and spread. And now here's the Hirschsprung mouse, just a normal Hirschsprung mouse with that spontaneous myogenic activity. Doesn't care that it got blue light stimulation. But in the Hirschsprung mice, where we transplant these cells, we get a little some diminution in the contractile response in the spontaneous activity. But the blue light stimulation leads to significant contractility, suggesting that those transplanted neurons are in fact now responding to blue light stimulation. So we know that the neurons we transplanted make connections with the muscle. So there's neuromuscular activity. That is blocked by tetrodotoxin. Notice you add tetrodotoxin, you basically are creating Hirschsprung disease because you're eliminating neuronal activity. So you get a return of your active myogenic activity. And the tetrodotoxin is still around, so the blue light stimulation now doesn't do anything. But the real question is, does it promote motility of the gut? To do that, we do spatiotemporal mapping. So if you put a gut in a dish, I showed you on the first slide, it will actually undergo peristalsis, and you can measure the changes in diameter of the gut and generate what are called chymographs or spatiotemporal maps. And this is a normal Hirschsprung intestine and a normal intestine. It's a Hirschsprung, but it's a wild type litter mate. You can see those contractions occur. They can be measured, and if anybody's ever looked at colonic motility tracings, you get the same thing. Here's the contraction starts and propagates distally, and you can measure the frequency, the amplitude, everything. In Hirschsprung disease, it looks different. You get this sort of random motion. There was one contraction there, it looked like, and then I think we see two over the course of 10 minutes here. And then when we transplant the cells, and here they were transplanted basically in the mid-colon, you can see some motility. You can see that it's primarily in the central region, but the frequency is increased. We see more of it. If you take those spatial temporal maps and rotate them, so now the contraction's starting here and moving distally in this direction, and I did that just to line it up to luminal pressure recordings. We can put a pressure transducer in the lumen of the gut ex vivo and correlate the contraction. It's not enough for a gut to have peristalsis. That peristalsis has to generate pressure to lead to movement of fecal contents and evacuation. And you can see that each of these contractions corresponds with a pressure spike. And that's the normal situation. In Hirschsprung, these two contractions were ineffective. There was no corresponding luminal pressure generated. And when we transplant the cells, these little propagated waves actually do correlate with some increase in luminal pressure. Remember, we're just injecting cells into one spot. So this is intended to demonstrate the capacity that these cells have to ultimately improve function, not to actually restore full motility to the gut. But this is all promising that the transplant of the cells can increase the number of CMMCs. These are the HAPCs of mice, the migrating motor complex. It can increase their velocity, the distance they travel, and the pressure they generate. Odd thing was that we noticed that our mice were living longer after transplant, and this gets to this whole topic of neuroimmune interaction in the gut, and the enteric nervous system has an important role in modulating inflammation in the intestine. That's why mice with Hirschsprung disease get enterocolitis, and humans as well. And we noticed that when we transplant the cells, instead of dying by 2 to 3 weeks, they were living about 10 days longer, and the severity of their inflammation was diminished by cell therapy. This is something we were hoping to follow up on. It was an interesting observation, and I think it's definitely worth looking into more. The problem is these mice still die, and that the fact that they die by a month and we transplant them at 7 to 10 days means we can only follow our transplants for a few weeks. So we needed a model that would allow us to follow them longer. So we generated a new model of aganglionosis in the colon. And we do that with the diphtheria toxin receptor. Humans have diphtheria toxin receptor, mice do not. So when we put a diphtheria toxin receptor on, again, Wnt1, all of the cells, all the neural crest cells in the body will express the diphtheria toxin receptor. If we inject diphtheria toxin into the gut, in the colon in this case, we cause aganglionosis. Those cells will die. And this is the ablated area that we can create. So we did an experiment where we take one mouse, or not one mouse, a lot of mice, but it's an autologous experiment. So we take a mouse and do a partial small bowel resection, and at the same time inject diphtheria toxin into the colon. So we want to make an aganglionic patch, and we're going to transplant its own cells back to the same mouse. So we do that small bowel resection, generate neurospheres. Here they express neuronal marker, glial marker, progenitors, and glia, because they're kind of the same thing. And we transduce them with a lentiviral GFP. We have to label the cells so we can find them because they're the mouse's own cells. So now they express GFP protein. And then 2 weeks later, because that's how long it takes to grow the cells and give them this lentiviral GFP, we transplant them back into that aganglionic segment. And you can see in this colon, when we take it out, the patch of aganglionosis is here. All the red is enteric nervous system. This is where the diphtheria toxin caused aganglionosis. And the green are the cells that we transplanted. And they set up a nice network of neurons and neural fibers that express, again, excitatory neurotransmitter, inhibitory neurotransmitter. And when we look at their ability to restore function, we get the same results we saw before. So now in this autologous setup, normal mice, electrical field stimulation, get a contraction. That aganglionic patch we created does the same thing as the Hirschsprung mice. It's got this spontaneous myogenic activity. We transplant cells, we lose that almost completely, and we restore a response to electrical field stimulation. And what we can see now is that the response at 3 weeks, which is what we were looking at with the Hirschsprung mice, because that's all they lived after transplant, there was some restoration of contractile activity. But in these animals, because they don't die from this, 8 weeks we could follow them, and we saw even more responds to contractile activity, which was really promising. We did the same with optogenetics and blue light stimulation. Again, you can see that the blue light in that aganglionic patch does nothing, but after we transduce these channel rhodopsin expressing cells, we get that contractile activity back. So we know that autologous transplants, autologously derived neural stem cells, can restore gut contractility. That response improves with time, but can enteric, like what we really care about, nobody sort of goes to the doctor, my contractile response is poor. I'm like having bowel movements is what they want to know. So can we figure that out? So the aganglionic patch we create does not cause constipation, and that's why the mice can live a full life. But this mouse has constipation. If you delete the nitric oxide synthase, the neuronal nitric oxide synthase neurotransmitter from mice, you get a model of neurointestinal disease. They actually have delayed gastric emptying and colon dysmotility. We transplanted these mice with red cells. Green is, they have an enteric nervous system, and that's green here. That's their endogenous one. And you can see that the red cells are transplanted in this region. All the green is their own endogenous. If it's also red, then it's our transplanted ones. And you see the transplanted cells end up like right in there, integrating together with the endogenous nervous system, which is something we couldn't really look for in Hirschsprung disease because there was no host enteric nervous system. So what we did in this model is inject either in one spot in the colon or create more coverage of transplant cells with 3 injections. The transplanted cells are red. Again, the green is the mouse's own nervous system, which lacks nitric oxide synthase. So nitric oxide synthase is an inhibitory neurotransmitter. It relaxes the colon. So we have to do these experiments in those non-adrenergic, non-cholinergic conditions. Normal mice, you can see that inhibitory response with electrical field stimulation. And then you get this rebound contraction. When you're missing nitric oxide synthase, all you get is the excitatory response. There's no inhibition. And when we transplanted the cells, not when we transplant once, but when we transplant 3 spots and get an increased area of coverage and probably more NOS expressing cells, we get a restoration of that inhibitory response. But for the sake of this experiment, what was more important was we looked at rectal bead expulsion and fecal pellet output. So rectal bead expulsion, your doctor never does to you. It's to put a glass bead in the rectum and see how long it takes for the mouse to evacuate it. And normally, let's say it takes 800 seconds. The knockout animal, much slower because it's constipated. And if we put in 3 injections, we get a restoration of that rectal bead expulsion time. Same with fecal pellet output. Instead of putting out 9 pellets, the knockout puts out 5. And after transplanting 3 at 3 locations, we get a restoration of fecal pellet output. So that this all positions us well now to we're in the process of sort of taking it to the next step so that we can get all the IND and FDA approval to move this on to humans. One of the challenges is we've been isolating the cells from the gut. And we can talk about why it's okay to take cells from the gut of a patient with a neurointestinal disease or Hirschsprung disease. But it turns out it is okay. However, it's a little bit invasive to get those tissues. We found that, remember that the enteric nervous system, the cells come from glia. Those neuronal progenitors are glia. Well, there's glia all over the body, and one place where there's a lot of glia is the Schwann cells along peripheral nerves. And one very innovative guy in my lab, Rian Stavely, found that in subcutaneous fat, there are neuronal fibers. And you can hear the neuronal fibers in red. But if you magnify this, you'll notice that the glial cells have actual cell bodies. The neurons are just fibers. Their cell bodies are elsewhere. But the Schwann cells are studying these peripheral nerves. And you can take those peripheral nerve fibers just from a chunk of fat. And by the way, we've made these cells, whether gut-derived or fat-derived, from mouse, swine, and humans of all ages. It does seem that younger is better. And if you take these nerve fibers, grow them as neurospheres, transplant them into Hirschsprung mice, he took a picture of this because it was a fecal pellet coming out of a Hirschsprung mouse, which we never see. And the electrical field stimulation, green, shows you the effect, the magnitude of the contraction generated in Hirschsprung mice after cell transplantation. So peripheral subcutaneous fat, turns out visceral fat is also very good for this, contains Schwann cells that have the same neurogenic potential when grown in the proper conditions in vitro, and even in the environment of the gut, they must be receiving the signals they need to undergo neurogenesis. The other source where there's a lot of Schwann cells is Hirschsprung disease. So I mentioned at the beginning, the aganglionic segment has no enteric neurons and no enteric neural crest-derived glial cells, but it does have those extrinsic fibers. There's these very thick nerve bundles, they're cholinergic. And so this mouse has Hirschsprung disease, it's an endothelin receptor B knockout, and all of the glial cells are green. And if you look at it, you can say, oh, look, there's normal ganglionic transition zone, and the aganglionic just has these fibers. As you zoom in on these fibers, you'll see that, in fact, those are cell bodies. Those are glial cell bodies. They're thought to be Schwann cell derived and come in not from the usual foregut to hindgut migration of neural crest cells, but along the extrinsic fibers. And so those cells can also be made into neurospheres, transplanted into the gut, and they will restore, again, the same. We transplant these cells, by the way, into that diphtheria toxin model, so into a patch of aganglionic gut, and you can see that they restore contractility in that gut. Also, that response is abrogated or eliminated, reduced, I'd say, by tetrodotoxin, again, confirming that it's neurally mediated. So now, instead of just having to harvest neural stem cells from the gut, we know we can get it from the aganglionic segment, which is going in the bucket anyway, or we can get them from subcutaneous fat, visceral fat. Turns out that bone marrow has nerve fibers along blood vessels, and those serve as a good source as well. So that's enough science. I spend a lot of time thinking about the surgeon-scientist thing because we found that over the decades, there continues to be a seemingly reduced maybe interest, enthusiasm, and ability for surgeons to maintain active basic science labs. That is not the case at Stanford in the Department of Surgery. It is not necessarily the case at some of the Harvard hospitals, but it is the case all over the country. And this is a statement made by Sam Wells, in 1996, that during the last several years, there's been great concern about the endangered status of surgical research. And the challenges are many. We definitely get increasingly asked to be clinically productive to generate revenues. And now people, you get out of med school and residency with so much debt, and the compensation plan shows you clearly at most places that if you operate more, you will make more. It's hard to get grant funding. I remember when I was a resident, a lot of the divisions of surgery at MGH funded their research off of extra clinical funds. Those days are long gone, and it's just getting harder. Work-life balance is much more important, thank God, than it used to be. But that does create added challenges to a career of being a surgeon scientist where time is like a valuable commodity. We did this survey in 2015 of all academic surgeons at the Academic Surgical Congress. We had over a 40% response rate. Over 1,000 people responded. And we asked, one of the many questions was, do you think it's realistic to expect surgeons to be successful basic scientists? This was only asked of people at an academic surgical meeting. And it was about a fourth trainees, a fourth residents, a fourth faculty, and a fourth surgical leaders. It was a good representation. That's division chiefs and department chairs, and 68% said no. We asked why, and the reasons were pretty consistent. It was the work-life balance, administrative duties, and clinical demands. And this paper got a lot of attention. It was published in Annals, and then there was a commentary in Annals. Nature Reviews, Urology picked it up, and the larger Nature picked it up as well. So the topic garnered a lot of interest, I think, because there's I hope, a recognition that surgeons add a lot to science. We see science, we see biology and pathology from a very different perspective. I go to a lot of basic science meetings, as I know some of you do, and it's very interesting. Or even when you sit at grant review sessions, the perspective a surgeon offers is not better. It is just different and it's not worse either, which some people often ask. It is different and it's valuable. And if we don't study surgical diseases, it's really not clear who will. I mean, Hirschsprung disease in our world, necrotizing enterocolitis, congenital diaphragmatic hernia, abdominal wall defects, they're very sort of surgical diseases because we see them, we treat them, we follow them forever. If we don't study them, who will? And also, I think that as academic surgeons, it's our responsibility to role model all aspects of our profession. So how to be a good surgeon, how to teach, and how to do research. And if we can't role model all of those, not each of us doing all of those, but as a department, as a division, having people who can mentor in each of those, then we definitely have failed our obligation. This is my favorite quote from Franny Moore, was the chair of surgery at the Brigham. He was a legend in like a lot of things, but fluid management or like electrolytes. He was all about very fundamental aspects of what happens to a patient while they're undergoing surgery. And he said, I'll just read it because we have a minute. The surgical investigator must be a bridge tender, channeling knowledge from basic science to the patient's bedside and back again. He traces his origin from both sides of that bridge. He's thus a bastard, and is called this by everybody. Those at one end of the bridge say he's not a very good scientist. Those at the other say he doesn't perform enough operations. It's harder to stay in the middle of the bridge than to retreat to one end or the other. But all of the fundamental advances in surgery, from Vesalius to Holstead to Cushing, have been made by those willing to maintain this uncomfortable posture. So I think to go back to the sciencey part, we definitely need to do better for the treatment of Hirschsprung's disease. I think enteric neuronal stem cell therapy offers a new approach. It actually treats the disease, which is usually a better thing to do. If the disease is missing neurons, then let's treat that, replace the neurons. We know that we can get neuronal progenitors, like I said, not just from mice, but from swine and from humans. The cells will migrate, they proliferate, they differentiate, they restore neuromuscular activity, and they improve colorectal function in the different models that we've used. And it could change the way we treat Hirschsprung disease and potentially other neurointestinal diseases. And I would say for all the faculty in the room, we need to continue to inspire, mentor, and support the next generation of surgeon scientists. These are the lab folks that did all this work, and without whom none of it would happen. This is my lovely family and my semi-lovely institution. I was driving around. Stanford is far more beautiful, I have to say, but this is what we got. So thank you very much.